So, good evening all, and welcome to the fifth and final session of Tree Health Week 2020 from the Arboricultural Association. My name's John Parker, I'm Technical Director at the Association and your host for tonight. I know I keep saying this, but it is always true. We've got a great lineup for your enjoyment over the next hour and a half or so as we conclude Tree Health Week by going into some real detail on specific pests and diseases and tree species with three expert speakers, Joan Webber, Pérez-Sierra and Greg Packman. First of all, my usual housekeeping, with apologies to those of you who've heard it all before. Please use the chat to say hello, as some of you are already doing, but please select all panelists and attendees as you do so. I'm talking to you, Andrew, and others. I particularly love to hear from you if, like Sophie and I, you've attended all 5 webinars this week. If you've got a question you'd like to submit, then please do so through the Q&A button so that I can keep track of everything. Don't ask your questions in the chat. You have up until the end of Sunday to take advantage of our very special Tree Health Week Fungi book offer, where you can purchase a package deal of our new books, one by Lynn Body and the other by David Humphreys and Chris Wright, for £70 plus postage and packaging for the pair. And if you buy both this week, you'll also go into a prize draw to win your copies free of charge and signed by the authors. We'll be doing the draw at the next live webinar in a couple of weeks. I'd also recommend you check out our website at trees.org.uk or via the links that will appear as if by magic in the chat to find out more about our online training courses and membership opportunities. I don't want to hold you to ransom, but the more members we have, the more of this sort of thing we can do. This week has been an exciting one for awards. On Monday, we announced that the winner of the 2020 Arboricultural Association Annual Award for Outstanding Contribution to Arboriculture is Jill Butler. You can visit our website to see the presentation video and the webinar that Jill delivered for the association a couple of months ago. Also on Monday, we enjoyed a great presentation from David Humphreys, who subsequently won the Individual Commitment Award at the London Tree and Woodland Awards, as decided by the marvellous London Tree Officers Association. And do please check out their website too, at ltoa.org.uk. And this evening you get to hear from another award winner, a multiple award winner, Greg Packman, who also picked up a gong at the London Tree and Woodland Awards, the Acorn Award for Community and Public Engagement. And if that isn't enough, Greg was also closely involved in the Urban Tree Festival, which won the Lockdown Award for engaging people in London with trees during lockdown. We really do only offer you the best here in Stonehouse. All of them are very deserving winners, and we appreciate not only their input into our own webinar series, but of course to the industry as a whole. And while I'm getting all weepy and emotional, I'd also like to say thank you to all of those who've been involved with this Tree Health Week, to our amazing speakers, Lynn Body, David Humphreys, Chris Wright, Mike Rout, Catherine O'Donnell, Lucio Montecchio, Alice Machugi, Vaibhav Barage, Nicholas Spence, Glyn Percival, Joan Webber, Pérez-Sierra, and Greg Packman. What an incredible list of speakers, all of whom freely gave their time and expertise to make this event such a success. I'd also like to say thank you to the team here at the Arboricultural Association, and particularly to our events coordinator, Sophie, who is the brains behind the operation and who's worked every night this week to make all of this happen. So thank you, Sophie. And of course, thanks to all of our wonderful audience. We've had more than 1,000 viewers from all over the world over the course of this week, and we're grateful to every one of you for your support. And there's plenty more to look forward to. We're not going to be doing a webinar next week, but we are getting it all up and running again from Wednesday, October the 21st, he says confidently as if he's planned a webinar, but there will be one, and we'll be putting on live events every week all the way through until Christmas. Ho, ho, ho. On to business. I'm very pleased now to invite your first presenter of our exciting triple header to the virtual stage. Please welcome Joan Webber.
And hopefully you can see my screen with my title slide, which says, what about Phytophthora? And before I start, I would just like to say a thank you for the invitation to be part of this event, because you really have had some great speakers, and I hope I'm not going to let the side down right now. Okay, so what about Phytophthora? Well, I think it's not a secret to a lot of people that I've been working on Phytophthora for a few years now, and very interesting it's been. But I wanted to start off just with some background on what Phytophthora are. Okay, I'm just having a little bit of a problem now, moving my slides. It was okay, but let's see if we can get to the next one. Okay, so Phytophthora. Well, I'm sure a lot of you will have heard about it. It's this genus of very aggressive plant pathogens. And if you're thinking, well, what Phytophoras do I know about? Of course, probably one of the most famous, or really the most infamous, is potato blight, Phytophthora infestans. And you can't help really respecting a pathogen which, in causing a disease on potatoes, then caused a mass migration of people during the potato famine in Ireland in the middle part of the 19th century. But if you're saying, well, that's potatoes, what about trees? Then what about something like Phytophthora cinnamomi? Phytophthora cinnamomi, known in Australia as the biological bulldozer, is on that list of the 100 most invasive organisms. And it causes disease, for example, not only in eucalypt forests in Western Australia, but if you go to the Iberian Peninsula, then you can see the impact that Phytophthora cinnamomi is having, for example, on the oaks there, holm oak, cork oak, and the amount of dieback that it's causing there. Well, we think about Phytophoras as being like fungi, and they look like fungi, and they operate like fungi, but they're not really fungi at all. In fact, they're much more closely related to algae or seaweed. And you can see that in some of their behaviors. They have these swimming spores, these zoospores, that are released into water and swim actively in water. And that's what you can see in the right-hand side of that slide. There you can see a sporangium that's in water, and the little zoospores that are in that sporangium have been oozing out of a little pore at the top of the sporangium and swimming off into the water. So Phytophoras need water to release spores, and they need water to infect as well. And of course, in Britain, we're all aware of several Phytophthora diseases that are highly damaging to trees, but confusingly, some of them are recognised as quarantine pathogens, and so there are certain regulations around reporting them, how infected plants have to be treated, whereas others are not quarantine pathogens, often been around for much longer, although some of them are showing increased impact more recently, but there aren't the same requirements and regulations around what you have to do if it's not a quarantine Phytophthora. But that doesn't mean to say if it's not a quarantine Phytophthora that it can't have quite a damaging impact. It's more to do with how long it's been around. Well, I want to digress a little bit as well and talk a bit about how many Phytophthores there are. And here we're really in quite a change in the situation. And I'm using a slide here that I've stolen from Clive Brazier, and he published this paper on Phytophthora diversity in 2009. And in that, Clive started off by showing really one of the first Phytophthores in the 19th century that was described by de Bary. It happened to be Phytophthora infestans, and how the number of Phytophoras that we're aware of has grown over time. So before about the 2000, we were aware of about 50 or 60 Phytophthores worldwide, not all of them affecting trees, but that was the total sum of our knowledge in terms of the number of species. Since then, the numbers that have been described or are emerging are just growing hugely. So now we're probably in a situation where there are well over 150 described Phytophthores. In fact, it's really a moving target. And this morning I turned to my research tool, Wikipedia, and asked it, well, Phytophthora species, how many are there? And it said 170. I'm not sure whether that's quite right, but it's a fair number. And Clive, at the time when he published this paper, said, okay, we might only know about 100 at this point, but there are probably another 400, 500, 600 out there that we don't know about at all. And I think that probably is the case as we're seeing more and more Phytophthores being described. And there are really a couple of reasons for why we're becoming increasingly aware of these new Phytophthores. And one is that we become much better at detecting them, and we have much better tools for isolating them, molecular tools for distinguishing them and identifying them. And the other is, of course, what has become quite an old chestnut now, the issue of trade. We are moving ourselves, we are moving goods, we are moving plants around the world. And as we do that, we give some of these Phytophoras opportunities to emerge from their native environment and move them to new parts of the world where they've never existed before. And when they are moved, that is when we become aware of them. So if I was giving this talk before 2000, I would have told you that Phytophoras were prevalent in nurseries. I would have told you that in the UK and Britain, there were probably 6 to 10 species of Phytophoras that were particularly damaging to trees, and it was especially ornamental trees that were affected by Phytophoras. It was really almost exclusively broadleaves where we worried about Phytophoras. And we also tended to think of Phytophoras as being root-infecting pathogens. And so they would attack the roots of trees, very often the fine roots, although this could extend into the root collar and up to the lower part of the stem. But the end result would be that you would see crown dieback. Giving this talk now, I would tell you many things very differently. I'd probably still say that they were prevalent in nurseries. And of course, nurseries provide such a wonderful environment for Phytophoras. There's a lot of watering, Phytophthora suppressive chemicals are often used, which don't necessarily kill the Phytophoras, but maybe suppress them. Some areas are using extracted river water for irrigation, which may bring Phytophoras into the nursery. And of course, there's a whole mix of plants. So plants are growing close by that normally might not be in proximity. And all this is very advantageous for nurseries. So that first statement is probably true. But I would say now we are dealing with far more species of Phytophthora that are damaging to trees. It's certainly not all about ornamentals, and it's certainly not all about broadleaves. And also, it's not just about root-infecting pathogens. We know that quite a number of the Phytophoras, some of the more damaging ones, are aerial Phytophoras. In other words, they don't attack the roots, they attack the above ground parts of the tree. That might be leaves, that might be shoots, that might be causing stem lesions, and really they may do little damage to roots. So everything is going on above ground in contrast to the root-infected ones where it's all going on below ground. So if I think about these pre-2000 Phytophoras, well, the ones I would have been telling you about would be Cinnamomi, Cambivora, Citricola, Cactorum, and Phytophthora syringae, one of the earliest also described. And then if I was thinking about some of the newcomers in the 90s, then I would be telling you about Phytophthora illicis, which attacks holly, and maybe Phytophthora olney, a hybrid which attacks alder. And you can see the emphasis is still on these Phytophoras, which we know attack the roots and are soil-borne, but we're seeing with some of them, particularly like Phytophthora illicis, here we've got an aerial Phytophthora as well. So the paradigm was still the same. But if we look at some of these post-2000 Phytophoras, then we've got things like Phytophthora ramorum, which is also known in the USA as the sudden oak death pathogen. We've got Phytophthora canovii, which we know on beach down in Cornwall. Phytophthora pseudosyringae, which attacks beech, larch, notophagus, and of course Phytophthora lateralis, which is better known for the impact that it has on Lawson cypress or Port Orford cedar in Oregon and California, but which we now have in the UK, and of course Phytophthora ostracedri on juniper, on a native juniper. So here immediately we see this shift really with a number of these Phytophoras attacking conifers, be it larch, be it Capiciparus, be it juniper, and quite a number of them being aerial. But the same story still goes on, that a lot of these are introduced Phytophoras. In fact, probably the majority of them are introduced, perhaps with the exception of something like Phytophthora pseudosyringae, which we certainly have evidence to show is native to Europe and may well be native to the UK. And just a little slide to remind you of the impacts of some of these Phytophoras. So there on the left-hand corner, you see dieback of larch up in Scotland caused by remorum. There on the right-hand side at the top, junipers being affected by Phytophthora ostracedri, really over a number of years there, because you can see some juniper bushes which are completely skeletonized, others which are still red and dying. And then down on the bottom right, we see these bleeds on the stem of beech caused by Phytophthora canovii. And then on the left-hand side at the bottom, we have at the very left these huge port-orchard cedar in Oregon being killed in their native range by this introduced Phytophthora lateralis. And there we see it in the UK on some ornamental Northern cypress, the same thing as port-orchard cedar in a park. And you also get a very different feel for how some of these introduced Phytophoras are spreading. So if you look at remorum on larch, you see it spreading aerially through these monocultures of larch planted in plantations, and you really see Phytophthora remorum affecting these trees on a landscape scale. On the Northern cypress in the UK, you just get ornamental examples of Northern cypress being picked off by this Phytophthora. And I should say for remorum and lateralis, these are quarantine Phytophoras. But probably what makes the difference between these two Phytophoras is, whereas with the larch and the Phytophthora remorum, you get huge areas of the landscape covered in larch. For lateralis and Northern cypress, you get individual trees affected in the landscape, and therefore probably much more in the way of barriers to the spread of this particular Phytophthora. That's probably the same with Phytophthora canovii down in the West Country, much more sporadic spread because of the fragmentation of the habitat and the way you will get individual beech trees dotted around in that environment, compared with something like the juniper in this area, where you get a lot of juniper in particular areas. If I move on to the next slide now, I want to talk a little bit about Phytophoras and the challenges that they particularly bring. And one thing that I will say that we see with their Phytophoras is their ability to change and adapt. And remember, they're not fungi, so they're not like ascomycetes that will be haploid, in other words, only have one copy of chromosomes. They're not like basidiomycetes that might be dikaryotic, for example. Phytophoras are diploid organisms, and that may help them when it comes to hybridizing as well. They get this mixing up in nurseries, and also these zoospores are little cells that don't have cell walls, they just have a membrane. So this may also help species, for example, to hybridize, where you get these zoospores coming together. I don't think Phytophoras are unique in the way they can cause asymptomatic infection, but you can see there on the right there a picture of an asymptomatic rhododendron leaf, no necrosis on that leaf, and yet we can still see those sporangia of the Phytophthora poking up from that leaf surface. And down below there, we have a picture of a chlamydospore of Phytophthora remorum in a root cell of a rhododendron. So they can be quite covert at times. Now, another thing that I don't think is just unique to Phytophoras, but it's something that we have to worry about with introduced organisms, that when they are introduced, very often people will feel that's a done deal. Well, it's got in, it's here, it's spreading. But what you may have is just a single clone of that particular species that's been introduced. But if you have repeated introductions, then you get different genotypes being introduced, and perhaps more ominously different mating types as well. So this gives the organism the opportunity to go through sexual reproduction, but also just to build up variation in the population. So more material for adaptation and selection. And the other thing about Phytophoras is some have very broad host ranges indeed. So something like Cinnamomi, potentially thousands of different hosts can be affected. Even something like Ramorum has a host range in the hundreds. And that's probably more usual than some of the Phytophoras like Phytophthora olney or Phytophthora illicis, which seem to have a much more limited host range. And then mobility. We so help Phytophoras the whole time by our behaviour. The way we move around the world, the way we move goods around the world, and the way we move plants around the world, and the way we disturb the environment that we have these organisms in as well. And of course, the big unknown, climate change as well. So there are these old and there are these new, and some of them will be native, some of them will be naturalized, and some of them will have been introduced relatively recently. And by relatively recently, I probably mean maybe in 10, 20, over 30 years, because it often takes maybe a decade or 2 decades before we realize that we have an introduction and we start to see the impact of that introduction. But using molecular markers and genomics is increasingly giving us clues about whether something is a native or whether it's an introduced population. And as a plant pathologist, we talk about what some of these indicators of an introduced or a non-native Phytophthora or non-native pathogen is. And so if it's non-native and recently introduced, we would expect it to have maybe limited variation. Maybe we'll see it only as one or a few clonal lineages. We might see evidence of recent genetic change as it's been moved into a new environment and come under different selection pressures, what Clive Brazier would call episodic selection. And most of all, the big kicker, if you like, we can see damage to some of our native hosts by these introduced organisms. And we wouldn't expect that if it was a native Phytophthora, because we would have expected that to have co-evolved with the native host and be in balance. But with these introduced Phytophoras, the geographical origins are really often obscure still, even for some that might have been around or introduced 50, 100, maybe even 200 years ago. And if we don't know where they've come from, then we don't know what their natural habitats are and what their natural hosts are that they've co-evolved with. And so we don't know what their optimal environmental requirements are, what limits them in their habitat. Importantly, we don't really know much about their hosts or really whether they're likely to respond negatively or positively to a change in climate as well. And maybe most critical of all, if we don't know where they come from, we don't really know how they've got here. Have they got here through the plant trade? Is it plant hunters who'd be looking for plants in exotic locations? And we don't know when they've arrived. So one of the things that people have been doing really, certainly in the last 10 to 20 years, is trying to find out more about the origins of Phytophthora, some of them very well-known Phytophoras to us, and do that by exploring areas where we think these Phytophoras may have come from. So I've just given you an idea here of where some people have been looking, and for example, we've had colleagues from the USDA Forest Service looking for Phytophthora ramorum in China, particularly in Yunnan province. And one of the reasons that people often look to East Asia is because that is such a centre of diversity of plants, and particularly rhododendrons, which many Phytophoras infect. Also, the Himalayas, remote forests in western Nepal. Here, this involved European scientists, including colleagues from Forest Research. Taiwan, to look for Phytophthora lateralis, And one of the reasons looking in Taiwan for Phytophthora lateralis was because there are native species of Camisiparus in Taiwan, but no sign of any Phytophthora lateralis disease there. And then more recently, and I'm going to focus more on this, explorations in northern Vietnam and also in southerly Japan. And for FR involved in these particular surveys, our emphasis really, or our aim, was to see if we could find some particular Phytophthora that are damaging in Britain and in the UK and in Europe, and these are Phytophthora ramorum and Phytophthora lateralis. One thing I can tell you about really all these surveys is you might not find the Phytophthora you go there to look for, but you always find a new Phytophthora, or usually many new Phytophthoras. Now, I've talked a little bit about Phytophthoras and how they're water molds and they have these swimming molds, and that is really one of the things that helps us when it comes for looking for Phytophthras. Because we don't need to go into these areas when we're on these expeditions and looking for Phytophthras and look for symptoms. Because remember, we might be going to a native habitat where these Phytophthras, the native Phytophthras, have co-evolved with a native host, and so we wouldn't expect them to be showing much in the way of symptoms. What we look for is these Phytophthras in river water or in water, and also in soil. Is there in soil maybe infecting fine roots? And we also have this opportunity with Phytophthras, and we often talk about this quite mysteriously, about baiting Phytophthras. But the great thing is that we've got a lot of water that we can sample and a lot of soil that we can sample, and we don't have to worry about looking for symptoms. So if we're baiting for Phytophthras, and for example, doing that in soil, what we do is we take some of the soil, we put it in a plastic box, we cover it with water, and if there are any Phytophthras in that soil, then the likelihood is that the spores will germinate, they will release these swimming spores, and they will swim up to the water-air interface in that little plastic box, and on that surface of the water, we float some leaves, which are particularly palatable or attractive to Phytophthora. And the Phytophthora spores will attack those leaves. So we can take a lot of soil and draw the Phytophthora out and look for the leaves that have any lesions on them. And by isolating from any lesions on those leaves, and you can see on the right-hand side there, we pick some leaves out, and if you look closely, you can see some little brown lesions on those leaves. That is the way we can isolate from Phytophthora when it comes to soil, by flooding the soil and letting the spores swim out and attack the new leaves that we float on the water. Or we can take the leaves to the water and do what I call their formal fishing with rafts of bait, floating our leaves in that river water, leaving them for a few days and then coming back, collecting them, looking for those necrotic lesions on the leaves and plating them out. Or we can be really lazy and just turn up at the river, pick up the leaves that happen to be there, hopefully ones that you think you're seeing this necrosis and it might be Phytophthora, plate that material out and you look for your Phytophthora in that way. And that has turned out to be a very successful way of doing a lot of sampling and really sampling over large catchments. If you think about how much water moves through rivers, so you can sample large catchment areas with water traveling through it, looking for the Phytophthora. Now, I said I was going to focus on some of the work we did in Vietnam and some in Japan, and I'm just going to start off by talking a little bit about the Vietnamese work. And this was done in conjunction with the Phytophthora Research Centre at Mendel University in Brno in the Czech Republic. Here, this is led by Thomas Jung. Many of you may already have heard of Thomas Jung. And also some other colleagues as well, Bruno and Tamara, and also Clyde Brazier from Forest Research, and done in conjunction with the Vietnamese Academy of Forest Science. And one of the reasons we chose northern Vietnam there is this Franzipan Peak there, which is a huge diversity of rhododendrons, and of course it borders China as well. So in sampling the rivers around that area, we were sampling waters that not only came from catchments in Vietnam, but also from China as well. And as I said, our particular interest in forest research are these Phytophoras that fit within this particular clade of Phytophoras, Ramorum, lateralis, which are closely related, but also some of their closely related cousins, Foliorum and Hibernalis as well. But we were also looking more widely as well. And so here we are, we're about to set off. You can see Clive, Thomas, Tamara and Bruno. And here we are aiming to go up on Fansipan, the roof of Indochina, as it's called. Now, some of you are thinking we're walking up there. We could have done that, but it would have probably taken us a couple of days. So we got the cable car up to the top of the mountain. That's the lazy way of the scientists doing it. And then walked down some of the mountainside and started sampling the soils there. But as well in that area, we sampled something like 16 forest rivers and streams, including Fansipan, which is 3,000 meters tall. And this just shows Clive and Bruno were doing some of that sampling. And we went into all sorts of different environments, whether they were agricultural environments, sampling riparian systems, mountainous areas, conifer forests, really looking at a whole range of different habitats, trying to make it as varied as possible to pick up as many Phytophoras as possible, using these baiting methods of soil and also of river waters. And as part of that work in Vietnam, we found 13 already described Phytophoras, 5 already known but not yet named species, and then 21 completely unknown taxa or species. So half of what we found hadn't been found before. We did find Phytophthora ramorum. Interestingly, we found both mating types. We do have Phytophthora ramorum as an introduced pathogen in the UK, but it exists here only as one mating type, as it does throughout Europe. We didn't find its cousin lateralis, though. We still don't know what lineage of ramorum or what lineages of ramorum there were in Vietnam, or really we're just starting to unpick that. But one thing that we could say is we never saw any plants that had symptoms caused by Phytophthora ramorum. We did look at plants, but we only ever found it in the water that we baited. So we can be pretty sure Vietnam is within the geographical center of origin for Phytophthora ramorum, but there we assume it's native and it is not damaging to the plants in those ecosystems. And so we have to ask ourselves, well, how has Europe and indeed North America ended up with Phytophthora ramorum? Could it have come from plant trade, via plant trade from Vietnam or even China, although ramorum hasn't been reported from China. For the survey work in Japan, we focused more in the southerly part of Japan. And here we were based at the Gifu University, working with the River Basin Research Center. Again, some really kind and helpful collaborators there. And we went to some of the different islands, Shikoku, Kyushu, Okinawa. Okinawa we were interested in, it's quite a bit further south, but sort of towards Taiwan, where previously Phytophthora lateralis had been found. So we were interested to know whether we would find lateralis in Japan and whether we would find ramorum there. It wasn't all hard work, I've got to admit. There were temples, there were beaches, there were really lovely meals in Japan as well. But there were snakes. There was a lot of time spent driving around, just as there was in Vietnam, getting from site to site, and probably some scrubbing around in rivers, which weren't such pleasant places to be. And a bit of comparing of samples, you know, my leaf is better than your leaf, really. Again, a wide range of different habitats explored here, maybe conifer or mixed conifer and broadleaf, dry volcanic terrain, mangrove swamps. Yes, you do find Phytophoras in mangrove swamps. And of course, because we are in Japan And because we know Phytophthora ramorum infects Japanese larch in the UK and in France, we went to Japanese larch stands as well. A similar sort of outcome to what we found in Vietnam. Some already described Phytophthora species, but many previously unknown taxa or species. In Japan, we again find Phytophthora ramorum and Phytophthora lateralis. And so Japan appears to be within the geographical centre of origin of ramorum and lateralis. But once again, we never found any plants that had symptoms caused by these species. And rarely did we see symptoms caused by other Phytophthora on this plant. And it also left us with the question, because we didn't find any species on any symptoms on Japanese larch, or indeed any ramorum associated with Japanese larch stands. For example, in the litter layer or in the soil, why is Japanese larch in Japan not affected by Phytophthora ramorum? And there's still ongoing dialogue with the plant health authorities in Vietnam and Japan on these findings. So it's a case of, well, what next and what are the questions? Well, there are still surveys going on in different parts of the world, although inevitably COVID-19 has limited this more recently. So areas that are being explored, for example, Borneo, Madagascar, and in the meanwhile, lab work is continuing to compare the ramorum from Vietnam and Japan with the lineages of Phytophthora ramorum causing epidemics in North America and Europe. Although, again, COVID-19 has also limited this. But there are interesting questions. How damaging are these native lineages of ramorum, for example, from Japan, from Vietnam, to larch? And of course, where else in this arc of forest from Myanmar through to Vietnam and China does ramorum and its close relatives exist? And indeed, what other Phytophthora are out there? And of course, we still don't really know from which of these areas are Phytophthora ramorum. By are, I mean what we have in the UK, and we have 2 clonal lineages of Phytophthora ramorum in the UK. We don't know where they've come from still. We're still working through that process of comparing them with what we found in Vietnam and Japan. And of course, the big question, is there more to come? So I think that brings me to the end, and I'll say thank you all for listening, and stop sharing my screen.
Great stuff. Thank you very much, Joan. As predicted, you obviously didn't let down the amazing lineup of speakers we've had. You were brilliant as ever. So thank you so much for that. Lots of questions coming in, which we're going to cover at the end, and some good chat going on. But first of all, we're just going to go through all 3 of our presenters, and then we're going to do our questions at the end. So I would very much like to hand over to our next speaker, Anna Pérez-Sierra. Anna, over to you.
Okay, then yes, thank you very much for inviting me to the last seminar of the Tree Health Week. Very exciting. I have been attending some of the seminars this week. I'm Anna Pérez. I work at Forest Research. I head the Tree Health Diagnostic and Advisory Service at Forest Research, and my topic for tonight is sweet chestnut blight. I have selected this topic because I have been working on this disease in the last few years. Then, before we start, I would like to say that this is a regulated organism. Cryphonetia parasitica is a quarantine organism in Europe, and UK is a protected zone. That means that we have special regulations that applies to importation of sweet chestnut material, and obviously these regulations are intended to minimize the risk of further introduction to the disease into the UK, and also there are regulatory requirements on importations of sweet chestnut material from non-EU member states. Then let's go to the fungus and to the disease. Sweet chestnut blight is caused by the fungus Cryphonetia parasitica. And this pathogen was first reported in 1904 on American chestnut, on Castanea dentata, and it was detected in New York. The disease then was named Chestnut Blight, and it killed millions of Castanea dentata, American chestnut, at that time. Then during the 20th century, Cryphonetia parasitica was introduced into North America and into Europe on infected chestnut plants. The first time that was detected in Europe was in Italy in 1938, and since then, the pathogen has been recorded in different countries in Europe. Then the pathogen is believed to be native to Eastern Asia, and the hosts are mainly Castanea species. Asian chestnuts have co-evolved with the pathogen, and they're more tolerant, where species like the American chestnut and the European chestnut, Castanea sativa and Castanea dentata, they are more susceptible. There are other hosts that may be infected by Cryphonetia parasitica, and some of the ones that are listed here, like oaks, maples, and European hornbeam. We have a few cases in Europe, but these cases have been only reported in situations where you have mixed forests that they are heavily infected with sweet chestnut, heavily infected by this fungus. Then just to show a map of the distribution, you can see it there. And let's talk about the fungus or the disease. The fungus is a bark pathogen. It is mainly infecting above ground parts of the tree. Normally, the fungus will enter the tree through fissures or through wounds. And stems or branches are girdled and die. Then above any girdling cankers, you will have the leaves will turn brown, wilt, and they may remain attached. Eventually they will fall, and you have dead branches. And if you have a very severe attacks, you can have just skeletons of the trees. The chestnut blight causes a canker. It affects the bark, and it causes a canker that normally is sunken or sometimes can be swollen. And usually below the canker, you have a lot of epicormic growth. And what I have done is I have prepared a series of slides with the symptoms, and we are going to have a look to chestnut blight, the symptoms, how to recognize it, the situation for the management of chestnut blight. And at the end of the presentation, I'm going to talk about the situation in England. Then let's have a close look to the symptoms. Here you can see the symptoms. You can see the first symptoms are this kind of orange discoloration in the bark. I don't know if can you see, I hope you can see my arrow, but you can see this kind of discoloration in the bark, orange or yellow, sometimes even swollen of the branches. You can see even the discoloration here. You can see it on the right-hand side as well. If you look for the cankers, they can be anywhere in the tree. And one tree doesn't have just one canker. One tree can have many cankers, and it can have cankers on the stem or on the branches. The cankers can be at any height, especially when you have young trees. The bark of the sweet chestnut is quite smooth. Then when you have the canker, then you have this kind of rough area where the bark is splitting and a change of the coloration of the bark, as you can see here. Then you have a canker here, you have the smooth bark, and you can see here rough bark again, and here is very clear. This one is at the base of the tree. Sometimes even when they're at the base, here at the base of the tree, they can be quite discreet. You may miss it because if you are walking on this direction of the tree, you will spot it. If you walk in the other direction, on the other side of the tree is nothing that lets you know that is a canker. Then sometimes when you walk in the woodland or in the forest, it's quite difficult to spot the cankers. For example, this is one case. Where is the canker here? Then when you have a closer look, you see that there, the canker is just there. Then sometimes when we go to do the surveys to search for this pathogen, we have to go with our binoculars to try to find out where the cankers are, because sometimes they can be quite high up in the canopy. Normally we see epicormic growth below the canthus. And this is, in winter, this is wonderful because the leaves are fallen, then you can see the epicormic growth and it lets you see through the canthus. Then, because you don't have the leaves, then you can see you have a canker here, a lot of epicormic growth in there as well. Here the same, you have the epicormic growth, you have it there. About the fruit bodies, it produces fruit bodies that are orange, bright orange in color, or sometimes more yellow in color, but you can see them here. Sometimes the bark is still quite smooth, although you can see it's a dark color, and you can see the fruit bodies in here or in here. You can see them in these pictures. And if you look, they are very, very small. They are like pinhead size, and you can find them on the infected bark or on the cracks or of the infected canthus. And when you have humid conditions, they exudate the tendrils of spores. And this is, you can see one here, there, or here, you can see them here as well. And the other thing you can see, one of the very classical or typical symptoms are the mycelial fans. And these mycelial fans, you will find them on the canthus below the bark. Then if you remove the outer part of the bark, sometimes these mycelial fans can be quite superficial, just under the bark. In some cases, will go quite deep into the xylem, and you can see here, and the lesions you can observe under the bark. Here is the same, we remove a panel, and you can see the brown discoloration tissue and the mycelial fans. And here you can see it again in this third picture. You see the mycelial fans growing under the bark. Then we have gone through the symptoms, what to look for if you are looking for chestnut blight, but let's talk about a little bit more about the fungus. Cryphonetia parasitica has two different types of spores, sexual spores and asexual spores. The sexual spores, we call them ascospores, and the asexual spores are conidia. In this case, both can cause infection. The ascospores that they are sexually produced are wind dispersed mainly from spring to the autumn, and they can travel longer distances than conidia. Conidia, they are asexual spores, and they're mainly splash dispersed by rain over short distances, maybe a few meters, or they are washed down the stem and branches. Then how do these spores enter the trees? We mentioned before, it enters through any fissures or wound, and any fresh wounds or any growth cracks in the bark, that will be sufficient for this fungus to enter the tree. Even any moribund wood tissue generated by drought, fire, natural competition, or cutting trees, it may also serve as an entry for Cryphonetia parasitica. We may have like birds, insects, mites, wind. They can transport the spores long distances. Obviously, plant material can travel freely in some cases, and that is another way of introduction of the pathogen into new areas. Also, recently it was shown that Cryphonetia parasitica infected abandoned galls of the oriental chestnut gall wasp, and this was detected in Europe. Then what about the control about this disease? Okay, we have seen the symptoms, we have seen it has different spores, how it infects the tree, but what can we do if we find it? Then chemical control so far has not been successful. Then the first attempt in any country is attempt to eradicate. And that, as you will see in the later part of the presentation, is what we have been trying to do in England. You can try to aim to do operations during the periods where you don't have spores. Then if you know that, we know that between, in the spring to the autumn is when you have spores, and you can time any operations avoiding this window to avoid colonization from the spores. Other options is breeding for resistance, but Cryphonetia parasitica is one of the few pathogens where we have like a good news story, and I don't know if we call it good news, but at least it's good news story, because we have what we call hypovirulence. And what happens is there is a virus that naturally colonizes the fungus and makes the fungus non-virulent. And this is being used as biological control. Then these hypoviruses are positive strands RNA viruses. They are located in the cytoplasms of the fungal host. Then Cryphonetia, there are different viruses or hypoviruses that have been detected on Cryphonetia, but not all of them have been shown to reduce the virulence of the fungus. However, the Cryphonetia hypovirus 1, or CHV1, that has received the most attention among these viruses because of its role on controlling the disease. Then the CHV1 induces a hypovirulent phenotype in Cryphonetia parasitica by reducing the parasitic growth and the sporulation capacity of the pathogen. Then if Cryphonetia is infected by the virus, by this hypovirus CHV1, how do we know? Then, for example, there is a way we can know in the lab when we grow the fungus in culture, for example, if the fungus is infected by the virus, is white in color. However, if the pathogen is not infected by the virus, is bright orange in color. Then, okay, this is how we can see it in the lab, but what about in the field? And in the field, if we have virus-free individuals of Cryphonetia parasitica, that means virulent strains of Parasitica, this one normally will invade the wounds of the bark of chestnut trees, will destroy the cambial tissues and cause the cankers that you can see with this sunken appearance. And cankers normally expand and girdle the stem and killing any part above of the canker. And these are the typical photographs that I showed you earlier on. However, if we have Cryphonetia parasitica infected by this hypovirus, it makes Cryphonetia parasitica non-virulent. And in this case, it can cause swollen superficial cankers because of the growth of the callus tissue, as you can see in the picture that just appeared here. They're known as healing cankers, and they do little or no harm to the tree, or this is another one of the healing cankers. If you want to read more about Cryphonetia, it's a wonderful review that was published in 2018 by some of our colleagues in Switzerland, and I would recommend that you have a look to that review. Then, okay, we have Cryphonetia parasitica that could have virus, infected by a virus that makes the fungus non-virulent, or we have the virulent part. But if Cryphonetia has the hypovirus, how does this hypovirus spread? Then when you have infected fungal individual, the hypovirus is only transmitted in conidia, not into the ascospores. Okay, that's okay. Then we need the next step is, but if we want to transfer the virus to another fungal individual, how does it happen? Then for this, we need to make sure that the hypha can fuse together. And basically, the virus can be transmitted to other fungal individuals via hyphal anastomosis. That means by fusing hyphae together. Then the hypovirus can be transmitted between isolates if they have the same compatibility type. That means that their hyphae are compatible and they can fuse together, and then the virus can also be shared. However, it's generally restricted on isolates if they have different compatibility groups. Then if you have different individuals together, but they are not compatible, then the virus cannot be transmitted. Then in an ideal situation, in your population, what you want to have is very low compatibility, vegetative compatibility diversity, because what you want is to have the virus and for this virus to be passed from one individual to another because they're all the same compatibility group. But unfortunately, that is not always like that. Then how do we know the compatibility group? Because I show you in the lab how we can see if the fungus has the virus, and that was one way to see it on plate, but normally we have a molecular method to see if the fungus has the virus. But also, how do we determine the compatibility group of the fungus? Because this is very important if we want to control it. Then here I'm going to show you a series of photographs. Here we are trying to see if this fungus, this individual and this one are the same compatibility group. This one and this one to see if they are the same compatibility group. And we have 4 different pairings in the plate. If they are compatible, their colonies fuse together, as you can see here. This is the reverse of the plate. And you see that no barriers are formed of incompatibility between isolates. However, if they are incompatible, you will see barriers forming where you have these ones are compatible, no barriers are formed, okay? Then, how is this biological control or CHP1 used in mainland Europe? In Europe, hypovirulence, as I said, the disease was introduced in 1938 in Italy, and I think it was in the 1950s when they realized this virus was present. And since then, obviously, this hypovirus has been successfully controlling the disease in many places in Europe. Hypovirulence is very successful, especially when you are targeting individual cancers and when you do this kind of applications intensively. However, as I'm mentioning here, sometimes you may target a cancer, but if the tree has different cancers, other cancers can kill the tree before it has an effect on the cancers, and is spread to other cancers within the tree. In contrast, the spontaneous spread of the viruses to new cancers in fungal population has had mixed successes. Then we have situations where even in the same country, in some regions, Cryphonetia parasitica has been controlled by the CHP1 virus. However, in other parts of the country, even that the virus is present, it has not been controlled. Then, although it's a good news story, it's a lot of caveats, if that makes sense, then, and it's not always to detect if we have the virus or not. For example, we know that the virus can be, in one cancer, you may have the fungus with the virus and the fungus with no virus on the same cancer. And this, we know it could be due to the incomplete movement of the virus within the cancers after infection. Another problem we have when we are looking or searching for this virus or hypovirus is that it's quite difficult sometimes to isolate from the healing cancers. And this could be due to the inhibition of both the fungal growth and the viral replication in these cancers. Then we know the CHP1 has been used or is used in Europe to control this disease in many places, especially is used in orchards and in under controlled conditions where you can access the cancers, or single trees where you can access the cancers. However, what about in the forest? And in the forest, this is a different situation, because what has been observed in the forest is you may have the virus present, the natural presence, and will be more successful than if you try to introduce the virus yourself. Then this is one of the potential problems that we may have. And for the virus to do its job, naturally, it may take many years. Then treatment of existing cancers with hypoviruses is effective, as I said, when you have a very well-managed orchards or plantations. But for example, in stands in North America or in European copies and stands in forests, it has not been effective. Natural spread appears to occur in some stands, as I said, of chestnuts in Europe. And even that you may have high incidence of the disease, the severity may be very low and very low mortality. However, in other occasions, it could be completely the opposite. You may have sweet chestnut stands in Europe that they have high disease incidence, relatively few healing cancers, but very high mortality. And you may have even other situations where healing cancers can be found in stands with high blight-induced mortality, having little apparent effect on the disease progress. Then I still want to think that these are good news stories, and the biological control is possible in woodlands. But as you can see, there are many different combinations and situations that we don't have clear explanations for them. Then, before I go to the situation in England, I just wanted to go very quickly just to mention, if we wanted to introduce the hypovirus into our woodlands to control the disease, it's not a straightforward process. You need to get a license for the introduction of a biological control agent or biopesticide. You need to do a pest risk analysis. You need to talk to the HSE and the Chemicals Regulation Division. Even if you want to have a permit for a trial, you have to comply with some requirements and EC regulations. This can take a long time. And I'm not saying that we shouldn't do it. What I'm saying is you have to start the process early to make sure that you can apply these biocontrols. But even when you have the permit, it's not a straightforward situation. This is a long-term solution, because the first thing when you have a outbreak of this disease is to determine the VC groups that you have in your population. Because as you know, if we have the virus, we need to transmit it from one individual to another one, and they should be compatible. Then you have to apply it in the field manually, and then that can take time. Not only that, you have to wait a season to see and evaluate what had happened in the field. And then you have to assess the cancers and also to check for the hypovirus present in the laboratory. As I said, this can be a long process. But all this introduction is because now I'm going to tell you about the situation in the UK or in England and what we are doing. Then the situation in England. If you want to follow the latest information, please check this website. You will have information about chestnut blight. And before I go to the situation in England, I have to say that all this work is being done in conjunction or in collaboration with the Tree Health Operations team at Forestry Commission and members of the advisory service at Forest Research. We work all together as one team, although we are different teams, but obviously the Tree Health Operation team is working in the field and we are working in the lab. Then the situation in England is that the disease was first detected in 2011 on trees planted for nut production. And this was in the middle of the country, and as you can see here in the map, and these plants came from France with their phytosanitary passport, and they were planted in 2007. The first symptoms were seen in 2011. It's a lag phase. The tree may be infected, but may not show any symptoms for a few years. In this case, after this finding, obviously everything was eradicated, and other cases were confirmed in the country on nurseries and new planting, and APHA did a lot of work on all of these surveys, and all these cases were eradicated. Then we thought the situation is finished for Cryphonetia parasitica. However, in summer 2016, we have a report from a single tree in Kent. You can see the tree here, a single tree The report came through tree alert to forest research, and it was Cryphonetia parasitica, and it was eradicated. Then we thought that was it. Then in December 2016, it was detected in Devon, the first detection in the wider environment and on semi-mature trees. These were the trees, and then they were eradicated. Then at the beginning of 2017, we found more sites in Devon and Dorset. And here are some of the sites, the infected trees, and you can see all the eradication that took place. Another site, another woodland, and you can see this is a coppice, and really sad as well, but all the eradication efforts to eliminate all these trees. Then in July 2017, the disease was confirmed in Southeast London. During 2018, then it was just, we were just doing, Forestry Commission were doing the service and we were testing the samples. And in 2019, it's been an ongoing activity with Forestry Commission and APHA, and new outbreak sites were discovered in in different counties. And this is another of the cases where it was a single tree. It was eradicated, it was eliminated, no other trees were detected in this area. Then what I wanted to show you is that basically we have been following up all the cases, and everywhere we could, we have been eradicating. And in some cases where it was just a single tree, like in this case, in some cases it was a coppice woodland, in another cases was a kind of a plantation, like single trees. Then in every case, we have been trying to eradicate. Then what we call the first survey is it ran between January 2017 and April 2018, where we have more than 100 locations with sweet chestnut that were surveyed, and they are on this map, as you can see. And of these over 100 locations, the disease was detected in 22 sites, and there are the ones that are black dots in the map. The clear ones are places where we had surveyed, but we didn't confirm the disease. Then we had in the southwest, in the Midlands, and the southeast. We looked for the presence of both mating types, and both mating types were detected, but usually one mating type was found in one site and another in another site. We only, in one occasion, we found both mating types together on one site, and this was eradicated as well. We have not found yet sexual structures of Cryphonetia parasitica at any of the sites where we have been working. And of all the collection of isolates we had from this first survey, we identified 11 compatibility groups. When I mention these compatibility groups, I haven't mentioned that in Europe is 74 compatibility groups that they have been recognized in different countries. We compare our isolates to European isolates, and 5 of our isolates corresponded to the already known compatibility groups. However, 6 of our isolates were unique compatibility groups, and they have not been found anywhere else in Europe. Then the question probably you're asking, did you find the virus on our isolates? Yes, we did find the CHP1 on 7 of our isolates from 3 different locations. However, the subtype of the virus that we found, it has limited hypovirulence. Then, although it's associated with Cryphonetia at these outbreak sites, it probably has limited impact on the virulence. It may be too early. Maybe we have to wait a bit longer until we see the effect. But from this first survey, one conclusion that was very clear is that the diversity of compatibility groups and the distribution at the outbreak sites, together with the finding of the CHP1, suggests that Cryphonetia parasitica has been introduced to the UK, multiple, in this case England, because we only found it in England, has been introduced multiple times through historical imports, because of course imports of sweet chestnut stopped in 2013. And just want to say thank you to DEFRA, who funded our research and still funding our research. Then what are we doing at the moment? Then surveys have been continued, and we are working on samples that they are collected by tree health officers or contractors or surveyors. And samples are always confirmed, not only by growing them in culture, but we have a real-time PCR method to identify if they are positive or negative. We have another method, another molecular method to identify the mating types of the fungus. We have another method to determine the compatibility group of these isolates, and we have, and all the isolates are screened for the presence of hypoviruses. And the person here in these pictures is one of our colleagues, Pedro, that is working on sweet chestnut day and night, just trying to see if we can find a way of controlling or find a biocontrol for this fungus in England. We are also looking to possible vectors of the disease. And just to finish the presentation, just to say, as I mentioned earlier on, we have Cryphonetia parasitica was found on abandoned galls of oriental chestnut gall wasp in Europe. And in the UK, as you know, in 2015, we have the first detection of oriental chestnut gall wasp. Obviously, I have been telling you about Cryphonetia parasitica, and eventually they have met on the same site. Then in 2019, we had confirmation of Cryphonetia parasitica infecting abandoned galls of the chestnut gall wasp in England. And this was published. You can see here, this is one of the galls, and you can see Cryphonetia and the lesion caused by Cryphonetia coming down on the branches. Also, we found different fungi that we didn't know we have in the country looking for Cryphonetia parasitica. One of them is Pneumoniopsis smithoglybii, and the other one is Dendrostoma luteum. Then if you want to find more information about chestnut blight, we have wonderful resources at Forest Research, also observatory. In fact, one of the observatory volunteers also detected Cryphonetia in one of the sites. And if you find any symptoms that I have shown in these photographs and you think could be Cryphonetia on chestnut, please report it through Tree Alert. Yes, you can find more information in some of these web pages, as I said. And thank you very much for your attention.
Thank you very much, Anna. We've got lots of questions coming in, and everyone's enjoying themselves in the chat. Thank you, that was really great. Oh my God, everyone's disappeared. I can only see me now. That's weird. Well, I feel like what you've all felt like for the last week now. Thank goodness, Greg's joined me. Okay, well, Greg, your presentation's like 3 minutes long. Yeah, is that what we agreed? Cool.
It's
so
very,
I'd
very,
now
very brief.
hand over to multiple award winner, Greg Packman. Greg, over to you.
Cool. Can you see the front screen, John? I'll take those, yeah.
Yes, I can. Yes, I can. Go.
Awesome. Right, so yeah, big thank you for having me on the, well, what's become the International Plant Health Week. And yeah, so I'm talking about Massaria, disease of plain, as I often do, really. So my sort of work with Massaria is I've been working with it quite extensively over the past sort of 5 years. Currently at my current job with Islington, but before that for several years at the Rural Parks. I've very much specialised in Massaria in the last couple of years, and as we'll see as the presentation goes along, it's a very different disease or condition, really, better word for it, than the two proper diseases we've looked at. So, brief overview of Massaria. So the causal agent of Massaria is considered to be Splunchnonomma platani, the fungus, and it was formerly called Massaria platani before they changed the name of it. And that original name is where we get the common name Massaria from. And it also operates in an anamorphic stage of Macrodiplodiopsis des Massariaceae, which I can actually spell from memory. I've had to type it out so many times, but they definitely don't make naming particularly easy or accessible. And it's a genus-specific fungi to Platanus. And it's an ascomycete fungi, as opposed to a basidiomycete or like a wood decay fungi. And this is one of the most important parts of the presentation. It's a weak parasite in its natural stage. It capitalizes on stressed, weakened, or preconditioned branches as such, and it's very much linked to drought stress and other environmental stresses. And in itself, Massaria isn't a significant tree health risk, but the presence and severity of the disease is very much linked to the health of an individual tree. So a bit on the biology. There's a bit of a chicken and egg scenario in terms of how Massaria is actually transmitted. Is it via spores? And the fungus produces an awful lot of spores, which can spread out on the wind very, very long distances. But there's also a general sort of school of thought that it's an endophyte as well. But in real terms, it's probably a combination of both. And ultimately, both require the same conditions in order to activate. So I think it's a combination of both, but in terms of practicalities, it doesn't matter too much. And on the whole, healthier branches tend to be unaffected by Massaria, and it sort of requires dysfunction of the wood in order for Massaria to activate as such. And that's quite often by reduced water levels from drought or dysfunctional wood created by wounding. And it's a weak parasite. So in the majority of cases, Massaria branches tend to be sort of smaller outer canopy branches. And by and large, we don't tend to really notice the smaller branches because we're looking for the larger, riskier branches. So on the whole, the majority of Massaria branches are ones that we wouldn't really notice. And the branches, they tend to be isolated individual branches. And as a general rule, in the majority of cases, Massaria doesn't breach the branch collar into parent branches, but as is always the case with trees, there's exceptions to the general rule. Continuing the biology, spores from Massaria are quite often found in large quantities on the upper side of branches sat towards the base of the branch. And Massaria is also classed as a strip canker fungi, which is a class of fungi considered to have the ability to penetrate bark and can colonize living branches. And the very early expression as such of Massaria is a canker or blister strip on the upper surface of the branch. It starts off with blackened spores, sort of gradually creeping, or want the better word, along the top of the branch. And it creates a strip of discoloration, sort of a variety of colours it's referred to as. And as the strip progresses along the branch, it kills the cambium beneath the bark before penetrating the wood itself. And it moves down the branch, but it moves around the circumference of the branch as well, creating like a wedge-shaped area of degradation within the branch. And after the cambium is killed, the bark itself flakes off and it exposes the discoloured wood, which in early stages is kind of like a darker cinnamony orange colour. And in much later stages, it's a much sort of grey, dull colour as it gets weathered. And here's some photos of the spores, kind of halfway between larvae and woodlouse, but that's what the spores under a microscope look like. And these are my photos. So the first one on the left, that's if you delicately peel back the top layer of bark off the Massaria branch, you can sometimes see these, the sort of black sporing or pycnidia surrounded by mycelium. And then if you're out and about, you see flakes off bark of a plane tree. Sometimes you can see the sporing structures on the bark as well. And the image on the left, here's a very early stage discoloured strip on a very small diameter branch. And then you can see this, you might need to peer in a bit to the screen. In the area I'm covering out by my mouse, you can sort of see dark raised blackened pustules from the spores. And then there's like a tiny tip here as well, kind of like the battering ram of Massaria as it sort of moves its way along the branch. And this is two photos of the same branch, but a much bigger example, much more established. You can see got that area of discolored bark, wood, large areas of bark flaked off. You can see it sort of tapers down further up the branch. Here's kind of a much closer, more clear example of the Massaria. And again, you can see that kind of arrow-like point of where Massaria just moves its way along the top of the branch before tearing off the bark and moving into the wood. Back to the biology. So once it's inside the wood, Splangenola preferentially degrades the S2 layer of the cell wall, sort of a cellulose-heavy area. And this is quite genius, really, on the part of Massaria, that it's able to shrink its hyphae into sort of thin perforation hyphae that enable it to go sort of cell to cell, rather than sort of battering its way through the front door, sort of sneaks in through the back door, through the cell lumen and border pits. And it's also able to move along via the rays and the vessels for a rapid spread through the wood. And it's this perforation hyphae that is one of the reasons why Massaria branches can develop as rapidly as they do. It's got a really clever strategy to expand while really not expending a great deal of energy in comparison to other fungi who would use a lot of energy to break down the sort of primary cell walls as well. And as the degradation starts from a tension wood, it gradually moves downwards. But in the early stages, when the tree still has good vitality, because when the Massaria starts, typically the branch still has leaves, it's able to contain parts of it's able to contain the colonized area for a time anyway. And that sort of gives the classic V-wedge that people are quite familiar with. And of course, it's a soft drop decay mode. And here's an example of that sort of V-wedge of Massaria. That's one of probably the best known symptom of Massaria from the very early days. Now, I think from early on, as in early on in our understanding of Massaria, this was considered to be sort of the symptom of what Massaria looked like inside the branch. I think in reality, what it actually is this is observed in the earlier stages where the tree puts down its initial barrier zones along the rays and then it contains it. But over time, it can breach these barriers and sort of spread around and down. And continue with the biology. So as Massaria progresses, the secondary walls are almost entirely degraded, but leaving the middle lamella largely intact. And it leaves the branch sort of devoid of cellulose to give it flexibility, but it sort of supports itself on a fairly rigid, stiff lignin framework. And as the wood further deteriorates, a series of cracks, sort of longitudinal and transverse, emerge in the tension wood. And the transverse crack, sort of running at a right angle to the grain, typically sits at the very barrier between the Massaria and healthy branch. And then as the branch goes on, the longitudinal cracks get much longer. And due to the reduced flexibility of the branch, the numerous cracks and the brittleness, the failure can be very quick and sudden. And all it really takes is sort of one big gust to come through, put too much of a load on the branch, and then branch snaps out. And in the early stages, at least, the compression wood is largely unaffected by Massaria and remains intact. And quite often, when branch failures happen on early Massaria branches, the intact wood fibers still support the broken branch. And I'll show you an image of what that's like. So, image on the left, you've got the Massaria area here, as you can see from this discoloration, and what was healthy compression wood here. So on this one, you can see all these jagged, buckled wood fibers and part of the branch tear here. So this was largely intact wood, although you can see what look like odd bits of mycelium going on. But this bit here, you can see it's very, very different. And this is a very even, almost uniform fracture surface. And this bit, it can snap off with almost no warning, with very little give. And this was a very, very large branch, actually, that broke out of the tree. So this is kind of where more of our concerns over Massaria can be aimed. And then the other image, that's just on a smaller branch, but a good representation of what those branch failures look like. Now, Massaria, as I mentioned, is a weak parasite. So in its natural state, Massaria is just a natural branch pruner of small diameter outer canopy branches, or worst case scenario, it helps shed weakened, shaded out branches that the trees don't really want to expend resources and function anymore. And if it was an oak tree in a deer park with that same type of fungi, such as Villaminia or Phytophthora, we'd be getting quite excited about these amazing ecologically beneficial fungi helping the tree shed off branches it doesn't need. And in its natural environment, I think that's essentially what Massaria is. It's just the parent species come from different areas, and for us, the tree that this fungus affects tends to be in sort of busier, more urban areas. But then the wood anatomy of the branch colour, the carbohydrate content of the wood, the vitality of the branch, as well as water and moisture content, typically restricts Massaria into isolated branches. And although you do get cases of Massaria passing back into the parent branch, on the whole, it's these factors that tend to keep Massaria isolated to individual branches. And I'll show you some photos on the next slide that kind of back that up. But on larger branches, they have a better ability to use their active defences to contain the spread of Massaria, and that can be for over a long period of time. And so Massaria doesn't initially and doesn't always function result in branch death. Quite often you can find a Massaria branch that still has leaf cover on it, still has foliage, because in the very early stages it's just a small area of Massaria that's being that's working within the tree. So this image here, kind of on the left, you've got the classic symptoms of Massaria branches. You've got that discoloured orange, you've got the flaked off bark, darkened bark and covered in spores. And over here, you've got the healthy branch. Now, if you look at the branch colour, you can see down the middle, there's a real distinct ridge and boundary between healthy wood, dead branch. And that's that barrier at the branch colour that prevents Massaria from spreading any further, in this individual case, into the parent branch. And over time, much like kind of with those same fungi on oak trees, the cambium in this branch colour continues to grow, and that branch will fall off, and then it will just callus and seal itself over. Perfectly natural part of the tree's existence. And here's a better example. I was quite excited when I found this branch because it was kind of exactly the reference photo that I needed. You've got all these symptoms of Massaria, discoloration, flaked off bark, spores, healthy wood, and then almost like a perfect line. It's like the tree's telling you where to prune if you need to. And yes, this is a great example of that distinction between the healthy wood, healthy tree, and the dead branch. It's a perfect visual guide to it, really. And here's another example. Kind of, if I have a favourite Massaria photo, then this is probably it, actually. All those classic symptoms, and then you've got the healthy parent branch, and you can see down here at the bottom the real distinction between dead branch, dead wood, healthy tissue. A really distinct line. So that's the barrier where the weak parasite can't go back into the healthier parent tissue, parent wood, as long as that's healthy. Also, you can see there's a distinct difference in the thickness of the branch collar compared to the wood. And that's because the cambium is still active and the branch collar is still growing. Now, is this weak parasite actually becoming a bit more aggressive? So the problem as such with Massaria is the rapid branch death at times and how it become brittle, and it's a risk issue for us rather than a big problem for the tree. And although Massaria has likely been around for as long as there's been plane trees, it's the risk issue that's become a more recent phenomenon. And there's a series of environmental and physiological stresses that create microenvironments in the branches that Massaria is able to take advantage of. So it needs those sort of preconditioned environments to take advantage of. So you've got your hypothetical healthy tree, good function, vitality, and water flow that's unlikely to lose Massaria branches from Massaria, but there has to be something that changes the environment externally to the tree that creates that environment for Massaria to get into and take advantage of. or if it's already within the wood as an endophyte, something to activate it within the tree. I don't know if it's kind of like an industry term or not, but it's something that I've always kind of mentally referred to as preconditioning as such. So there's a whole variety of factors that can impact tree health, vitality, water flow, and moisture content in the tree. So this is basically like a non-exhaustive list. And the one most associated with Massaria has always been drought stress and dryness. And it's not just a lack of water in the soil, but it's the tree's ability to uptake water as well. So if you've got compacted soil or contaminated soil, then that can impact the tree's ability to uptake water. I put primary pathogens because that could be something like anthracnose or mildew, or something more severe that can create stresses in the tree. Air pollution isn't doing our trees any favour. Wounding and injury as well. But ultimately, it's just things that create dysfunction in the healthy tree, that disrupt the water flow, that dry out the wood, that enable these weaker parasites, pathogens to come in and capitalise on that stressed area. And something that I'm quite curious about is the impacts of the way that we're pruning trees. Does that actually benefit Massaria and do more damage in the long term, as well as the interaction with other fungi? So here's an example of what I consider to be like a preconditioned branch by pruning. So you can see here's the pruning cut on the branch, and you've got a necrotic strip running down, and this is that same branch. But above the pruning point, all seems to be healthy wood. And that's sort of showing the classic symptoms of Massaria, and that's running in a strip, tapering down from the pruning point. Now, most of what you read about Massaria is that it tapers out from the branch collar and down the branch, whereas it's now tapering in from the pruning point and running backwards to what it's supposed to be doing. And that seems to be an area, sort of quote unquote preconditioned, if you will, by pruning, that's created a favourable environment for the fungus to capitalise on. And hopefully you can make this out as one branch in a sea of branches, but this branch here, so the branch was cut off here. I can't remember why. This is one of my old trees, but I can't remember why we pruned it. And if you look closely, you can see you've got that blister strip from Massaria, that cinnamon discoloration, running back from the pruning point, runs all along here, and it sort of stops around here. And if you look at this part of the top side of the branch, it's all seems to be healthy wood. So that's kind of Massaria running backwards, if you will, from the pruning point, rather than tapering out from the branch collar. And that seems to have been taking advantage of that area of dysfunctional wood. The moisture contents really reduced, the water flows stopped, and it's just been able to activate. And it seems to be that area of dysfunction that's creating these environments for Massaria to capitalise on. Again, here's another example. You've got a twin branch, and there's that pruning point, and you can see closely there's a darker, older strip of necrotic bark here. And I can't in good faith say that's 100% the reason, because there could be any number of reasons. But what I can say is that the way that that's been pruned, it has at least enabled the conditions in what may have otherwise been a healthy branch. It's created that initial dysfunction, and it's now changed the environment in the branch. It's enabled it to become more susceptible to Massaria. Now, whenever I sort of issue and specify work, I'm very rigid in that I don't want large pruning cuts up to the trunk. If there's no pruning point, I'd much rather them leave a stub that can be stable rather than a big open wound on the trunk. In the British Standard, it gives a recommendation of no more than sort of 10 centimetre diameter pruning cuts, which I try and stick to. But in this instance, you're creating large areas of dysfunction on the trunk of the tree. And if we were talking about habitat and ecology work, then fantastic future habitat. But when it's kind of risk management of urban trees, I want them to be as healthy as possible. I want to try and avoid as much area of dysfunction as possible. Plus also in this instance, you have a sizable area of dysfunction on the top of the branch, but actually quite a large area of the branch was unaffected. So it's one, I'm never going to say we should stop reducing trees, because that's kind of me committing career suicide and a bit nonsensical. But it's very much a case of think about the implications of what happens to the tree when you're recommending reductions or branch removal for Massaria. To remove the immediate or more visible risk, are we actually then creating the conditions that make things more challenging in the future? So that's kind of my personal preference, is at all costs, there are unavoidable times, but I try and avoid as much as possible creating large pruning wounds to the trunk. It takes a long time for them to seal over. Now, a healthier branch, healthier tree, it can actually contain the spread of Massaria for a time through its active defences. So the third wall of Coda stops or contains the radial spread of the branch, and the spread down the branch is attempted to be contained by wall 1, which is the weakest barrier, and that seems to be why Massaria is able to spread far more quicker along the length of the branch than the circumference. And it seems to be dependent upon the tree's vitality to how it can maintain these barriers, because it's an energy-driven barrier. So as an example, the first year you could have an effective barrier which can contain the initial spread, but there could be a drought or something similar in the second year that enables the barrier to be breached and Massaria spreads, and it could be some time again before the tree has enough energy to put down a new barrier. And for larger branches, it can take several years to either die or fracture. So here's an example of the barriers, Massaria barriers as such, and there's a bit of a callus there. And in this example, a bit clearer, you've got this sort of squiggly line barrier separating healthier wood from Massaria. This image here, it's the same branch, just zoomed out. So you've got the main barrier here. Also, if you look, you've got an older barrier here and here. There might be one there, can't say for definite, but if you look very closely, you can make out just here, it's a different shade of grey and some straight lines. So I think, however many years ago, the first colonisation of Massaria was here and contained. Then the following year, there may have been a drought, knocked the tree's defences, the fungus got out, colonised this area, then the tree put down a new barrier here and here. Then same thing, another drought may have happened, and then the fungus got out again and spread here. And what did it for this branch in the end is that I put it down for removal. Had it stayed in the tree, there could have been a new barrier here, and that bit could have become grey. Who knows? So it seems to be, but it definitely is the case that for a time, if a tree has enough energy to do so, it can put down barriers to contain the spread. It just needs to maintain that spread. Maintain that energy. And here's an external example. So you've got the darker area here, that sort of first, as a personal preference, I don't like using the word infection for Massaria. To me, I don't think it's the right term. Other people may disagree, but that's my personal preference. So you've got the first sort of colonisation or area of Massaria here, and you've got the callus from the barrier, a bit of woundwood, and then it sort of breached it and gone into that second area here. And in this example, potentially 1, 2, 3 different areas based on the discoloration. So there's potentially 3 different barrier breaches on that example. So one thing that's been quite interesting, not just with Massaria, but our general understanding of fungi as a whole, is that there are far, far more species in any one part of the tree going on at any one time than we used to realise. And Splanchnonym has always been considered kind of the causal fungus in Massaria. But recent research in Europe has found other fungi, Haplocystis beckelii, in Massaria samples. It's much more well known in Europe. And then I found a dead plane tree that had Massaria symptoms, got some samples, and there was no Splanchnonym, just Diplodium mutilla, which is a very obscure fungus. I don't know too much about it, but that really surprised me that there was no Massaria in the Massaria sample, just a different fungus. Neophysococcum parvum is becoming identified more frequently in plane trees. That has some implications on tree death, but it hasn't been any case in the UK from what I know, so I won't go into that too much. Metaporia mediterrana, there's more of that going on as well. Innonatus, there's an interesting relationship between Innonatus and Massaria as well. And hopefully this is one that we don't get at all. But there does seem to be far more going on in terms of different species of fungi interacting together. So is it really a complex rather than a specific disease? Because I tend to separate Massaria, the condition, away from Spongianoma, the fungus. So conclusions really. The first and most important one to keep in mind is that Massaria is a weak parasite that capitalizes on stressed and weakened branches. And it's a natural branch pruner in its natural state. And it needs environmental and physiological stresses to really capitalize on. But as plane trees age and environmental stresses increase, instances of Massaria may well increase as well, or they probably will in real terms. But also, the way in which you think about removing Massaria branches, consider the potential consequences of how the pruning operations may impact the tree for future health. By removing the immediate risk, are you creating problems for the future? If I look back at the work I've issued in the past, I've definitely made things worse on a number of examples, and that's something that I try and learn from. Also, what's the health of the individual tree? Can it be improved? So street trees will be a challenge, but parkland trees it could be. And in theory, and again, this isn't real terms, but in theory, the healthier the tree, the less likely that Massaria potentially will colonise large branches. And this is just a collection of references that have been very useful for Massaria. And that's all. Thank you.
Thank you very much, Greg. Always a pleasure to see you and listen to you. That was absolutely brilliant. If we could get Joan and Anna back on video as well, that would be great. Because we have 3 presenters tonight, we're running a little bit later than we normally do at this stage, so we're going to sort of rattle through some questions relatively quickly. So probably don't submit any more, folks. That's the first time I've ever said that, but don't submit any more questions. It's going to start with a question for Joan. Here's Joan. Perfect. Right. So thank you all very much, first of all. That was all brilliant. And I don't know if you've been looking at the chat, but everyone's thoroughly enjoying themselves. Joan, a question, first of all, from Andrew. Does felling of infected trees with Phytophthora spread it more, or does it control it over time?
I think that entirely depends on what Phytophthora it is. So if it's Phytophthora ramorum and say a larch tree, then the sporulation goes on the foliage, on the needles. So you want to take it out of that environment. So by felling it, you remove that tree so it's not lofting inoculum into the air, and you get it on the ground, and then you can get rid of the tissue that would sustain sporulation. If it's a root-infecting Phytophthora, cutting that tree down won't make any difference at all. You would probably have to grind out the stump, but even so, I think you would do a great deal of damage, cause a great deal of destruction, probably spread a lot of inoculum around. So I think sometimes it can be a requirement or a good thing to do if it's an aerial Phytophthora. Other times, I don't think it's a good action at all. And I think the other thing is we assume that all the Phytophthora is focused on that tree, and we often don't think how widely it's already there in the environment. So I know people have worried about things like Cactorum or Plurivera, Citricola, if you like, and said, well, we need to remove these trees that have infections and we'll control that Phytophthora. But I think they're quite widespread in the environment. So I think it can be an option, the right option, depending on the Phytophthora. But a lot of the time, I don't think it is the right option.
Thank you very much, Joan. So it all depends by the sound of it. Anna, question for you. We're probably going to get through about 2 questions for each one of you, by the way, so just to be prepared. Anna, I've been keeping notes of my questions. Here they are. Right, a question from Ian. Thank you, Ian, by the way. You're one of several people who've been submitting very good questions throughout the week. So thank you so much for your contribution. Ian says, and let me try and get this right, Anna, are the sweet chestnut blight vegetative compatibility groups competitive with each other? Could a compatibility group that is susceptible to the hypovirus be used to displace the others? You're muted. You're still muted, Anna. That's it.
Okay, thank you. Yeah, sorry. Different things about the compatibility groups. The compatibility groups, first of all, it's not something that is fixed. We keep finding new compatibility groups as we go along. As I said, there are 74 recognized in Europe, but every country has their own ones in a way that haven't been found in other places. Obviously, when we find one of these compatibility groups in one area, normally it may be one, or normally you find only one compatibility group in one area or in one tree, then it's like they colonize their own space. But the virus it will transmit to the same compatibility group. It's not like it goes better for one or for the other. If you have the virus in one of the compatibility groups, in theory, it will transmit to the same compatibility group. This is always in the lab. However, the beauty of this is in nature, apparently, the transmission of the virus, it can also happen between different compatibility groups. However, when we work in the lab, we are trying to force a situation. And obviously, we know in the lab that is more difficult, but in the field, it can happen.
Thank you very much, Anna.
A few different things there, I don't know.
No, that's good. I should say again, I say this every night, we've only got time to go through a fraction of the questions that have been submitted, but we always make sure all of the questions and all of the chat are sent to all of our speakers. And obviously our speakers are very, very busy, but they'll be able to have a look through the questions. And if they feel they particularly want to answer something, then they'll be able to do so. If you want to follow anything up else, then please email me, john@trees.org.uk, and I can ask the questions. So don't panic if we don't get to you. Thank you, Anna. Greg, a question from David. Is there a possibility that rising nitrogen oxide or ozone pollution levels could be particularly important factors in increasing Massaria occurrence? NO2 being known to be a central cellular module involved in plant drought signaling. Does pollution make it worse, Greg?
Yes. So from what I gather, the nitrogen oxide is one of the worst ones on plant health. There's a series of part of the environmental stresses is basically from air pollution as well. I forget what the exact component of air pollution is, but it degrades the cuticle on the individual leaf, which of course is a protective barrier, and makes things a lot worse. So I can't go into an overly complex explanation because I don't know a complex answer, but in short, I think yes, it is quite a significant potential problem to make plant health worse from air pollution.
Thanks, Greg. I don't know if Joan or Anna have any thoughts on that one at all.
Not for me.
No. Look at that, Greg. It must have been perfect. It must have been perfect. Okay, thank you very much. Joan, I'm going to combine two questions for you. One from Anthony, so I'll take a deep breath. Thank you, Anthony. An obvious question for Joan after Glyn Percival's revelation yesterday about populations of penicillin endophytes in some trees combating other pathogenic fungi. Are there any identified fungal or bacterial antagonists of Phytophoras? Especially as Dr. Webber, you've made clear, in their genetic heartlands, there are often no trees with discernible symptoms. And I'll just take this, that's Anthony's question. And Andy's asked a similar question, but a bit shorter. Are there pathogens that attack Phytophthora which may limit their effect in their native habitats?
Those are very good questions. I can't really answer them other than to say, in culture, certainly, bacteria are Phytophora's worst enemies in many ways. And I can see Anna Perez is smiling at that because she too works on Phytophoras as well. So I'm sure there are these checks and balances in the natural environment. And also it's thought that, for example, mycorrhizal fungi will protect roots and root tips from attack by Phytophoras. It's just something that we know very little about. And because we're often don't even understand where these Phytophoras really come from, we're not in a position to explore what these natural interactions are in that native habitat.
Thanks, Joan. Anna, did you have anything to add to that one, or is that no? Great stuff. I love the fact that this has happened quite a few times, particularly during this week, but people are asking really intelligent questions to which our panelists have to sort of say, well, we don't know yet. It's just so exciting about the research and the work that's going on. It's all very exciting, isn't it? There's so much we don't know. Okay, Anna, a question for you. Ricardo put this into the chat, but I'm going to go with it anyway. Are there different effects of the virulent and non-virulent sorry, it's very, very late. Chestnut blight on the physical and mechanical properties of the wood? Do the virulent and non-virulent affect the physical and mechanical properties in different ways? You're muted again, sorry.
Sorry, I keep forgetting. It must be Friday, yes, late. Yes, as I said, this is a bark pathogen, and it mainly affects the bark and the cambium, and very rarely goes to the sapwood. Then if, as far as we are aware, unless you have the virulent, obviously, it may go a little bit in some of the layers of the sapwood, but normally it's a wound pathogen going to the bark and the cambium. It won't affect the sapwood.
Okay, thank you very much,
I don't
Anna.
know if, Joan, you have something to add to that, but I don't think we have any, yeah.
No, I don't think we really have anything else that we can say about that. Yeah. I mean, all that with the hypervirulent strains, they appear to allow the bark to heal, or they're not damaging enough, so the tree can counteract their effect by callousing.
Thank you both very much. Right, Greg, I'm going to let Anthony have another question, because I do want to raise the point that Greg's far too modest to say it, but I think one of Greg's most important contributions to this whole debate is that for a very long time, everybody just accepted the truth that you could only see Massaria from above. You could only see Massaria from being in the tree. And I know that from looking at far too many plane trees, I think, Greg, you're aware that you can actually see it from the ground as well. Anthony said that you've spoken elsewhere, Greg, about the fact that you can see Massaria from the ground by various signs. But has drone technology any practical use in this scenario to be used instead of climbing if you do need that diagnosis from above?
Yeah, I mean, I've never used a drone. Most of my work on Massaria has been in Westminster, which has a no drone zone policy. And you would have to fill in, I think, like a license for every single tree inspection, which for me would be thousands. So it was never a practicality for me. I know that in Milton Keynes they're considering it because they've got thousands and thousands of plane trees. And then a guy that I was at college with did his dissertation on drone inspection of Massaria. I don't know anything else beyond that because I haven't seen him in a very long time, but there's definitely potential there. I know that if I were to do it, I would be the guy that gets the drone stuck at the top of the canopy within 5 minutes or so.
You really would be.
Yeah, I think as long as you're a good drone pilot, you know what to look for and how to operate the camera in a way that won't miss what you can see from either imagery or on the ground. And I think if you can make use of it, then absolutely it should be something that's not ruled out and something that is looked into more.
Well, I suppose a lot of your work has been, demonstrating that really you shouldn't need that above ground view of it. You should be able to get those signs from below in most cases. Yeah,
I mean, I always advocate the ground-based as the starting point, but I don't doubt that there will be situations where the most practical use of time and efficient use of time could be a drone. So if it can be done, I'd never rule it out, but I'd say start the ground-based and work on ruling that out before. doing anything else. Yeah, I think there's potential, definitely.
Thanks very much, Greg. Well, look, I just want to wrap up with one last question. And it's a question for me. This is our final bit of Tree Health Week. I should probably have prepared you all for this, but I'm going to go with it anyway. I'm interested to know, do you feel sort of positive about the future? We often hear these horror stories about tree health and these diseases that are coming in. They're going to rampage through our urban forests and through our countryside. Overall, and I'm not going to hold you to this, but do you think the future is quite bright? Do you think we should be positive? Joan, I'll start with you. How are you feeling about the future?
Honest answer, I'm quite worried about the future when it comes to our trees. But that's also partly because, as I said, when we have some of these introduced organisms, we often don't know. They could be insect pests, they could be Phytophthoras or other pathogens, but it often takes us quite a long while before we know they're there and having an impact. So if we're assuming that some of the things we're dealing with now were introduced quite a while ago, maybe some decades ago, even if we've seen a ratcheting up of a lot of the regulation and plants that come in and out, I still think we'll have a legacy to deal with. So yes, I am concerned looking to the future.
Thank you, Joan. Anna, I'd ask you the same question. As the person who has to manage the Tree Alert email address, how do you feel about the future?
I'm a very positive person. No, I cannot listen. There are moments when you feel really depressed, it's true, but also the trees are very resilient, and I believe in that as well. Then I think we will lose some species. It's as simple as that. But for me, maybe we will have others. Then, yes, I think I'm a kind of a hybrid. I can get very depressed in some times because you think, it's very sad to see these trees dying and going. But at the same time, I think the trees, it's the genetic of the trees and trees' resilience to what is coming. And it's so much we don't know about. Then for me, I'm going to keep a little bit positive. Come on, it's Friday after all.
Thank you, Anna. Okay, well, I guess the deciding vote goes to Greg then. Greg, how are you feeling about the future of tree health in the UK particularly, but everywhere?
I often get told that I'm a very pessimistic person, and I'm not overly hopeful on a lot of things. I don't want to end it on a negative, but the immediate threats look really bleak. Not just from pest and disease, but from climate, from development. But there's more and more awareness of the importance of trees. There's things like this going on. There seems to be, compared to 10 years ago, the level of knowledge and accessibility for people to actually do something to try and prevent it has skyrocketed. So I'm very hopeful on that. It's just whether we can get the political support to actually improve things remains a big question. But yeah, the immediate future doesn't look great, but I do think we're in a far better position than we ever have been to actually try and affect positive change, and I hope something positive can come from that.
Thank you, Greg. Well, I think that's a suitably positive note to finish on. Thank you so much to the three of you. That was just brilliant, so enjoyable and so interesting. After a week of this, my head is just I've just taken in too much, but thank you so much. You're all great. I guess I can finish on a positive note, which is what gives me real hope for the future, is the engagement of the audience of the webinars all week, the questions of positivity, the international relationships that have been built up, the fact that we've got all these amazing presenters who've donated their time and experience and knowledge to help us put on this thing. It's just been really remarkable and very special. And thank you to the three of you. Thank you to all of our speakers for the whole week. And thank you to all of you out there, to the audience. We really do appreciate it. Please, if you want to get in touch, visit the website, trees.org.uk. Please feel free to drop me an email, john@trees.org.uk. And like I say, there's no webinar next week, but the following Wednesday, we will be starting our next series. And I hope that we'll see you all then. So please take care of yourselves, look after each other and everything, and have fun and enjoy a weekend. and we will see you very soon. Thank you so much, everybody. Good night.
Thank you. Bye.